This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 457, recorded on September 1st, 2017. This episode of TWiV is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with your first purchase with free shipping by going to blueapron.com/TWiV. This episode is also brought to you by the Defense Threat Reduction Agency. Part of the U.S. Department of Defense, the agency's Chemical and Biological Technologies Department hosts the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Find out more at cbdstconference.com. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me here in New York City, Dickson Despommier.
Hello, Vincent.
Where the weather's quite nice.
It's one of the most uplifting days of the year that I've experienced so far. Clear,
21 Celsius, mostly sunny.
Yeah, it was actually pure sunny when I woke up, but it's partially sunny now.
I'm really sad that the humidity's gone.
Are you really?
I love humidity. I really do.
Well, I'm fine without it. Thanks.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here.
Welcome back.
Thank you. Yeah, I missed it.
You did good things, right?
Yeah. Did some really, really cool stuff. Went and saw the eclipse, and it was awesome, as we were just talking about before the show.
And last Friday was your birthday.
Last Friday was my birthday, yes.
Happy birthday.
Thank you.
To you. Very good. And we hear that you are now older than Dickson.
No.
We'll talk about that later.
Maybe
in behavior.
Is your weather like ours?
It's gorgeous. It's
gorgeous.
Just beautiful, almost completely clear skies, 68 Fahrenheit, 20 Celsius. Just a lovely late summer thinking about autumn type of day.
That's right.
I'm sorry. I just love the hot, sultry days of July. I just love them. I don't know why. I really do enjoy them. I like stepping outside and feeling that humidity. Now I step outside and it's cool. I really don't want it. I'm going to find a place.
I like maybe 2 or 3 days like that in the summer, and then I'm done with it.
I like it all the time. I could have it 365.
Really?
You should move to South Carolina, where I just was for the eclipse.
So it's hot and humid all year?
I don't know about all year, but it was blazing.
I was in Tennessee, and I was experiencing the same thing.
Actually, you should talk to Rich.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
It's not humid in Austin, is it?
It's a little humid, but nothing like Florida. Yeah, if you want it sultry all the time, I can recommend Florida to you. But you want to go somewhere where there's Spanish moss growing in the trees.
Oh,
well, you can't. That's impossible. Where there's Spanish moss growing in the trees, because that's the test.
Yeah.
If there's enough moisture in the air to grow Spanish moss, you're there.
You had that in Gainesville?
We did indeed, in spades, but we do not have it here in Austin, Texas.
How about Savannah, Georgia? I bet they have it.
Oh yeah, you got Spanish moss there.
All through the South. Mississippi has it. Georgia has it. South Carolina has it.
New Orleans, that's where I'm from.
Yeah.
All right.
And so, wait a minute.
What's up? Go ahead.
Come on.
What do you got?
It's 90 degrees, which is 32 C, and it's sunny.
You had some rain though this week, right?
Some rain.
Fortunately, we got 10 inches of rain and a lot of wind over the weekend. Last weekend, it cleared up. It stopped raining on Monday and cleared up completely by Tuesday. So we were lucky. We dodged a bullet. I hate to. It's just painful to think about what others are going through.
Yeah.
Yep. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, everyone.
Hey.
Sounds like I have exactly the same weather that Alan does, and it is just gorgeous here. I'll take the fall the whole time. You can have my summer days, Vincent. I'll take the fall.
Okay, I know that most people do like this fall weather, but I just happen to like the humidity. Brianne, I passed you, I think, twice last weekend, bringing my daughter. Well, we did 2 college runs. We went to Marist on Saturday, then Sunday we went to Ithaca College, and we take 24 out.
You drove right past us.
Yeah, I drove right past you, yeah. To, what is it, 287, I think.
Mm-hmm.
287 north to 17, etc. So I waved.
That well.
I waved, but you didn't notice. You were just bent over the microscope.
That sounds about. Actually, I was bent over the syllabus prep this week.
Syllabus prep. How did your class go?
Both of my classes have been great all week.
Nice.
I have 2 wonderful classes full of students.
To tell them that you were on a very famous radio show.
I did, yes.
Excellent.
They were very excited.
That's great.
Did
Mostly
they
the virology students.
No.
All right, Dickson. I've had this article.
Yes, you have.
For you for a couple of weeks.
Right.
And now you're finally here.
I am.
It's a Nature news article. First genetically engineered salmon sold in Canada.
Right.
This has nothing to do with viruses, but I just thought it's in. I know you like the fish.
It's interesting. Yeah, well, I like to eat fish too.
And it took 25 years to bring these fish to market.
Yeah.
They are basically, they put in a growth hormone gene from another fish.
Extra growth hormone gene.
All right. So they got a growth hormone gene from Chinook salmon.
Right.
So what are these salmon? What kind are they?
These are Atlantic salmon.
Atlantic salmon. So
That's
they
right.
put Chinook. So are they bigger Chinooks?
Chinook is the largest of the salmon, yes.
And they put genetic regulatory elements from the ocean pout.
Yeah.
Do you know what a pout is?
I do.
Not my pout, but
No, it's a bottom feeder.
why would they use
Just
genetic?
sits in the corner all day long and shrugs.
Maybe they just had them from that particular fish.
No. I think they have been working on pout for a while.
So this makes a low level of growth hormone all the time.
Right.
So the fish grow
They do.
quicker.
They do.
And that, of course, gets them to market more quickly, right?
Exactly.
So what's the normal time to get to market for a salmon, do you know?
Which salmon are we talking about?
Atlantic.
Well, it depends on how big you want them, but I mean, they'll go out to sea and come back 2 years later.
Do you think these are farmed, though?
Oh yeah, they're all farmed.
These are farmed.
Sure, these are all farmed.
And they say that one of the advantages of this, which I don't quite understand, is that you can farm them inland in tanks. Whereas I guess that salmon farming ordinarily happens in
the ocean,
like penned places in the ocean.
Right. Yeah, and they use them in the ocean, but you can do this in freshwater too.
These were actually
you could
raised.
do that even without the genetic modification.
Correct. Absolutely. The biggest drawback is the waste that they generate. And in places like Norway, where they have these pens and fjords, and it's a very, very friendly environment because you get no storms and things like this to disrupt your salmon farming, all the waste goes straight down. There's no current to take it out to sea and to dilute it. And it just piles up on the bottom of the ocean where these salmon are being raised. And then eventually fish diseases start to take advantage of the situation. And there's a lot of infectious pancreatitis and things like this in salmon as the result of over farming in certain areas.
So presumably, if you could rear them in a more controlled environment, you could take care of that.
If you go to Canada, near Vancouver, there's a huge current that goes between Vancouver Island and the mainland of British Columbia. And when you raise salmon there, all that waste just gets swept right out to sea, and there's no problem. There's a big movement, however, I must say, I just got back from a meeting in Squaw Valley. Had nothing to do with salmon fishing or anything else, except that the guest of honor was David Suzuki. And David Suzuki is a very famous communicator / biologist / geneticist. He's now in his middle 80s. The way he describes himself, he says, I've just entered the death zone of my life. And he was being very friendly and upbeat about it. He says, everything dies.
So what did he feel about these?
He hates fish farming. He hates the idea of it. And he's got lots of reasons that he gives as to why he doesn't like it. And he's actually incurred the wrath of the entire fish farming industry of Canada as the result, because he's a very famous Canadian. He's a scientist and he comes out against it. But I wish I had told him, I said, David, don't you think that if they hadn't farmed Atlantic salmon, all the wild Atlantic salmon would have been extincted now because they're delicious. I mean, they really are very good to eat. And they wouldn't stop netting them just because there are fewer of them.
I have a picture here of these fish at the same age, twice the size of.
Yeah, but you can do that by selective breeding also. Yeah. You don't have to insert more growth hormone if you don't want to. And,
well, I think these are even more rapid growing than ones that are selectively bred.
Sure.
A company called Aqua Bounty Technologies from Maynard, Massachusetts.
Yeah.
Developed these fish. They have sold the first 4.5 tons to Canada, but they won't say to who. It took them 25 years to get to this point. The fish were raised in Panama, and they plan to make other sites. Panama.
This is a cold water fish
in tanks,
we're talking
in
about.
Tanks. To keep them cool.
So
I don't know.
far down.
Well, then they're going to do one in Prince Edward Island. They also want to do one in the U.S.
Right.
But here in the U.S., they have some problems. A law for the 2017 U.S. government budget includes a provision that instructs the FDA to forbid the sale of transgenic salmon until it has developed a program to inform consumers that they are buying a genetically modified product.
Right.
Senator Lisa Murkowski from Alaska, who inserted the provision, has called the salmon fake fish.
Yeah, well,
because they compete with the Alaskan fish industry.
Just think a little bit about that.
This is all about the money.
Sure it is.
Sure it is. And there was the industry. The industry's also got a bit of a black eye right now because there was just a big rupture of a salmon farm pen out in, I think, Washington State. That released a whole bunch of Atlantic salmon out there, and they're trying to catch as many of them as possible because they're not a native species there. And that, of course, raises the question, why are you farming a non-native species in the middle of the water where they can escape in the first place? But
one of the big fears that the sports fishermen had was that the salmon, the farm salmon, which are weaker and less fit for life in the raw, so to speak, will interbreed with the wild salmon and produce hybrids which are less fit, and therefore it will affect the salmon runs for the fishing, sports fishing. So that hasn't happened yet. That has not happened. And these salmon are so unfit that they wouldn't even recognize a wild salmon if they saw one. So there's little danger of that happening, but nonetheless, the fear has been raised, and people do respond to those fears.
I was wondering how these growth hormone adapted engineered salmon would do in the wild.
They not well at all because they're overfed. That's the thing. If you keep feeding them, they'll keep growing. But of course, wild salmon have to forage for their food, and most of those salmon end up above the Arctic Circle near Greenland and in between Greenland and Iceland, and they feed on krill. That's basically what they're eating. And these salmon are fed pellets. They wouldn't recognize a krill if they saw one.
If these were in the wild, they would grow so quickly they would need a lot of food, which they could not get, you're saying?
And it's possible that they would die as the result because they'd starve to death. They don't know how to hunt for food.
That's the bigger problem is that farmed salmon in general and farmed trout and all the farmed fish have no experience in their brain about how do you hunt a bug.
No survival skills.
And if you're fishing and you're catching farmed fish, you throw the bait in the water and they bite it immediately because to them something falling on top of the water is how they get fed.
Vincent and I took a float trip out west when we were at TWiV at Hamilton, and all we saw were wild fish. There were no and you really had to respect them for their ability to distinguish between a fly that looked exactly like what was hatching and a fly that doesn't look exactly like what was hatching. They will not bite at the fly that doesn't look exactly like.
Pretty discriminatory.
Very discriminatory.
Dickson, who eats salmon in the wild? No, not people, but animals, other animals.
Bears.
So the question
everything
is,
bigger than salmon.
If a bear ate this, if one of these were in the wild and a bear ate it, would it now grow huge because of the fish growth hormone?
This is fish hormone, not bear growth hormone. This is fish growth hormone.
Doesn't bind the receptor. Okay.
No, it would be destroyed by eating it because it's a peptide. You'd digest it on the way through your digestive tract.
Yeah.
Wouldn't survive.
No, I'm talking about if you eat the fish, you're going to get the DNA for the growth hormone.
You just that too.
And you will take up DNA, and who knows?
No,
No, it doesn't work.
Don't think so.
Whatever
We're
you
not
do,
transgenic
don't eat DNA.
animals.
Right, don't eat DNA.
That's right. Whatever you do, don't eat the nuclei.
So you're saying there's no horizontal DNA transfer among organisms in
because
the
when
wild?
you eat something, it passes through your digestive
I'm not so
tract.
sure about that. I think the bacteria could take it up in your gut and maybe pass it on to
What's
you.
the
Who knows?
Then we've already
what's
been
the evidence
exposed
of
to
that?
all the different
Yeah, I agree.
genes
I want to
that
see
are-
that.
That's where we're going here. I don't think it's
Exactly.
an
No,
issue.
do you know how many cells we shed every day through our gut tract?
Yeah, lots.
Yeah. So why doesn't the bacteria then below that take it up?
It's not useful to them.
It doesn't.
Well, they
do.
do. They do take up human DNA for sure. Absolutely.
Really?
Yeah, no doubt about it. And we take up bacterial DNA. In fact, you have it
Yeah.
in your brain right now, Dickson. No, I mean, one of these guys here said they should have done an environmental impact study. Here's a
Can't do it.
Fish grown in tanks, right? I guess you have to say, well, if one got out, what would happen?
Oh,
Right.
everything requires an environmental impact study.
Even in a tank?
Mm-hmm.
No, you know what would happen? The seals would probably eat them because they're easy to catch.
Sure.
They're so domesticated.
I don't have any problem with this, but why? Last point here. Do you think it's necessary to label this as genetically engineered fish?
No.
Mhm.
No.
No.
Everything has been genetically
Yeah, I
engineered,
agree.
just not quite in this fashion. But no, I don't think so.
I don't think so either. But many people feel that way, obviously. Or maybe they
People-
don't. They just
people-
want to protect
Yeah,
their
people who have a vested interest in the fishing industry or people
That's
who've
right.
been sold the
That's
bill of
right.
goods by them.
All right.
Exactly right.
Okay, Dickson, hope you enjoyed.
People also feel that when they eat something that's been genetically modified, they're going to get sick. What the hell does that idea come from? I have never in my life thought of anything so fallacious as
It
that.
just comes out of propaganda.
It's just awful. Yeah, it's fake news. That is definitely
Yes.
fake news. And that's scare tactics. And you're right. Absolutely right. It's totally unethical to talk like that.
All right, we have a snippet which comes our way from Kayam, who writes, Genome-wide analysis of influenza viral RNA and nuclear protein association. He sent this paper to us. Also, I'm super excited for This Week in Immunology. Thanks for all that you do.
A new show.
So that's a new show, but it's not going to be called This Week in Immunology. It's going to be called something else, which we haven't decided yet. Decided to break the mold, which we already did with urban agriculture, right?
We did.
So I decided that this would be a cool paper to do. The subject is interesting. It's an interesting assay. And the author, the senior author, this is a paper published in Nucleic Acids Research. And it may be the first NAR paper we've ever done here on TWiV. I don't remember ever doing one. Now, NAR was a journal that I used to look at all the time in the old days, but not much anymore. Anyway, the first author is Nara Lee, and the senior author is Seema Lakdawala. And this comes from Microbiology and Molecular Genetics at the University of Pittsburgh School of Medicine, where I visited not too long ago. And I put a picture of Seema in there in the show notes with her scientist Lego set.
That's great.
And
It's a nice set. I have it too.
you have that one?
Lovely.
I do.
It's called the Research Institute, right?
Yes.
She had it on her desk. She was very excited about it. And we did a previous paper of Seema on TWiV 363. She had been a postdoc with Kanta Subaro, and we talked about her work on influenza virus replication in the soft palate. Anybody remember that paper?
Yeah, I remember that.
I think so.
So that was Seema's work. She since went on to the University of Pittsburgh, where she's a new assistant professor. So this is cool. This has to do with influenza virus. Now, negative stranded RNA viruses, their genomes are typically coated with protein. Dickson is frowning.
No,
They're coated with protein, nucleoproteins, or NP or N protein, flu, VSV, paramyxoviruses, they all, in contrast to most plus-strand RNA virus genomes, which are naked, like polio. Not all of them are naked. The coronas are not naked, but most of the others are. And so people have been interested in how these nucleoproteins associate with the viral RNA. And for flu, structures of the RNA complexed with the nucleoproteins have been solved by cryo-electron microscopy. And as they say in the introduction, the universal depiction of these RNA protein structures, which they call RNP, ribonucleoproteins, in textbooks is of a uniform random binding of nucleoprotein over the entire length of each vRNA segment, like beads on a string. I've actually included in our show notes a picture from our textbook, Principles of Virology, which, in fact, this is a picture solved by cryo-EM, so you don't actually see the RNA, but you can see that the proteins are arranged in a helical fashion, and they've modeled in the RNA in these reconstructions, and the RNA seems to be completely coated with nucleoprotein. So hence the idea. Now, in this paper, they want to address that. Is that true? Or are there regions of the RNA that are naked? So the procedure they use is called HITS-CLIP, HITS-CLIP. High-throughput sequencing of RNA isolated by cross-linking immunoprecipitation. HITS-CLIP.
HITS-CLIP.
You basically take influenza virus particles and you irradiate them with ultraviolet light, which will cross-link the protein with the RNA. So you fix the nucleoprotein onto the RNA genome right there in the particle. And then you extract the nucleoprotein RNA complexes, you digest them lightly with RNase, and then you immunoprecipitate these protein RNA complexes with an antibody to nucleoprotein. You can then label them with P32, run them on a gel, and purify them away from the uncomplexed RNA, and then sequence them, do deep sequencing. So what you're looking at then is since you're immunoprecipitating with antibody to the nucleoprotein, you're looking at sequences that are protected by the protein.
Protected from the RNase.
Protected from the RNase, and they're brought down, they're purified by immunoprecipitation with an antibody to the protein. So whatever RNA is stuck to, you sequence it, you can say, here is where the nucleoprotein is binding. So it's a clever way to do it, right?
It is really clever.
Yeah.
One of the things I like about their methods is that the cross-linking step is done just on virus that's in culture medium.
Right.
So they haven't done anything to pure. That's as native virus as you're going to get, I think.
That's right.
All right.
Yep. And the paper is nicely described, very clear, and lots of good controls.
And it's open access.
Open access.
Yeah.
And you actually get...
I really liked the controls myself.
Tons of controls.
I like that they diagrammed the method.
Yeah, that too, which is
You know.
you would not be allowed to do in a high-profile journal.
This is a segmented genome, though. Is that correct?
It is segmented.
So how do you know which segment you're looking at?
By the sequence.
Okay.
Because they're going to sequence these RNAs eventually. And you can say, that's hemagglutinin, that's neuraminidase.
Okie doke.
How many segments are there, Dickson?
6. As I recall?
8.
8.
How many genome types are there in the viral world? 7, yeah. I'm confusing you with numbers, right?
No, that's all right. I'm usually confused.
They did this procedure with 2 different influenza viruses, the WSN strain from 1933, an H1N1, and a 2009 H1N1, which is the newly emerged pandemic strain at that time, and they do it multiple times. And what you find is that the profiles of NP-RNA binding are similar. They're not the same between the 2 viruses, but they're quite similar. But NP binding is neither uniform nor random.
Aha. It's not a series of beads.
It is not.
It's very poorly strung beads.
Yeah.
Poorly strung beads. Hey, it's a good title. Poorly strung beads.
Because we have such a shortage of titles for this episode.
Yeah,
indeed.
really.
We have dozens. So that's basically the result, that nucleoprotein doesn't cover the entire RNA. There are bits that are uncomplex, and they feel that this is dynamic, most likely, that there are pieces of RNA that are covered and not covered, and they happen to catch it at a point where certain RNA sequences are not covered. So that begs the question of why, right? So maybe you need to have exposed RNA for packaging, for reassortment, and so forth.
So my question before, though, related to, if you're looking at this just through the cryo-EM method, and you've got 8 segments to this genome, can you sort them according to their morphology? Or do they all look exactly the same until you start sequencing?
I think by cryo-EM, they look similar, but they're not exactly the same. I looked at the paper last night, and they had multiple panels for the different segments.
So you could possibly then start to arrange the genome according to which segment is there just by looking.
Well, you could, of course, they're different sizes, so you could.
I haven't looked at the cryo-EM, but one of the problems is that these are not just beads on a string, but folded back on each other, right?
Yeah, they are folded
on
back,
themselves.
Yeah.
Nice.
So it's a more complex structure, and one of the things that the authors point out in this is that the techniques that are used to do the reconstructions from cryo-EM, they're not doing single molecule stuff; they're doing averaging.
That's right.
Because they're taking a bunch of molecules and averaging. And so if there's, in particular, if it's dynamic, and I suppose, Dickson, you've got a point, if you're mixing up segments, can't differentiate one from another, it's all going to average out so that it looks like beads on a string.
Yeah, but then when you start to sequence, aren't you going to get dominance and not so much dominance according to who's present in the solution when you start to sequence it?
Well, they cross-link it, so you fix the NP and the RNA. Then you ask, what does the NP protect? RNA,
I see. You're
right?
just sequencing the exposed regions.
No, you're sequencing the protected.
Yeah,
the exposed sequences are digested away, right?
Yeah. Okay.
So we mentioned the controls, which are cool. They basically hybridize oligonucleotides to regions which should or should not, they predict should or should not be covered with NP, and then treat it with a ribonuclease that will digest the hybrids and see whether it's cleaved, and it supports the notion that that the protected regions are in fact protected. This made me wonder whether you couldn't do an experiment where you just take these things native, no cross-linking or anything, and just tickle them with ribonuclease, or maybe you could cross-link them and just tickle them with ribonuclease. You ought to be able to get fragments out that makes sense relative to this packaging pattern. It
depends how dynamic it is, right?
Yeah.
If it's changing rapidly, you may not get any reasonable result from that. You might digest everything, because at some point in time during the digestion, even if it's 5 minutes, it might be enough to hit everything.
So during viral replication, though, this has to all be exposed somehow, right? Mm-hmm. So what happens to the protein? How does that work?
So what happens is when the viral RNAs first get into cells, they're covered with nucleoprotein. Yeah. The RNAs are each associated with a polymerase molecule, which begins to make messenger RNAs. Okay.
Each has it. Okay.
The messenger RNAs go on to make protein. Those messenger RNAs are simply made for protein synthesis. You have another set of RNAs that are full-length plus strands.
Right.
Those are copied to make full-length minus. All right. And now, as soon as there's enough nucleoprotein made in the cell, those minus strands begin to be coated with nucleoprotein. And so you have a shift between mRNA synthesis and genome RNA synthesis.
Another question. If this is all a pool of these genome fragments being replicated inside the virus factory, how does it sort according to fragment 1, 2, 3, 4?
Some people.
And in fact, I think that's a key issue in the whole paper. I think that's one of the places where this is going.
Do you have a lot of defective virus produced during influenza?
I think something like 1 in 400 are infectious. So you do have a lot of defective, and some people have argued that means it's just a random pulling of 8 segments from a pool. But there's more evidence more recently which indicates that there are conserved sequences at the ends of each segment that are unique, and that direct packaging of the correct complement into virus particles. And you can swap those sequences among segments and get those packaged as well. So people are still working on this, but it probably isn't random. It's probably sequence dependent. But here,
so those sequences are those sequences actually at the end? Could some of these exposed sequences participate in that as well?
Sure, because I think some of these are at the ends as well, right?
So and they say that in the discussion. That's sort of something they're hypothesizing.
Right.
So per cell, how many active viral particles are produced during an average infection?
Probably thousands.
Thousands or
tens.
So that means what an enormous
It's a
energy
big pool.
waste producing all of those extraneous pieces.
That's the viral lifestyle. You make lots of offspring, and not all of them succeed, right?
And it's not your energy that you're wasting; it's the cells.
Sounds like the U.S. economy.
It's called a parasite, right?
That's right.
So yes, that's one of their ideas, as Brianne said, that maybe these exposed regions are involved in packaging and reassortment and so forth. So you can imagine that they're going to look at that.
So do viruses that have single-stranded DNA or RNAs, they are more efficient at producing particles?
Not necessarily.
Not
There's other
necessarily.
things that can go wrong.
Yeah,
you know?
sure.
not
Okay.
just
All
just
right.
packaging.
All right.
You know, there are other viruses that are equally
Wow.
inefficient, if you want to put it that way. Although so we're talking about the particle to PFU ratio here.
Right.
That's right.
In some, it's almost 1. Some viruses make very good
very-
particles all the time.
very
Others
efficient.
do poorly, and we don't really understand that.
What is it in flu? Do you know, Vincent?
I'm thinking 1 in at least a few thousand. I'm going to look in the textbook where I have a table.
I mean, you didn't commit that to memory.
I remember it being pretty low.
Let's see.
It's
that would be
It's pretty low for polio too.
that would be in the genomes and genetics chapter, which I happen to have written. Here we go. I have a little table here that talks about the particle to PFU ratio of selected animal viruses. You can tell I'm scrolling. Okay. And it's really instructive, so we're going to definitely bring that up. No, that's genomes and genetics. Is that chapter 3?
Yeah, it's chapter 3. That's Monday's lecture.
Maybe you already know
I've got
the answer.
I've got
then,
it
Brianna.
here.
What is it?
Influenza virus 20 to 50 particle to
That's not
PFU
bad.
ratio.
That's not bad.
Yeah, I got
That's
it.
actually not
table,
bad.
Table 2.2.
Polio virus is 30 to 1,000.
And how many viruses have segmented genomes? I mean, of all the viruses that you know.
How many is tough because it would be what we know of, right?
Yeah, of known viruses.
They're not huge.
Not-
They're-
not
think so.
that many.
I didn't
not
think
that
so.
many.
And
By
the
the way,
advantage here is that you can mix and match when you
Yeah.
get inside of a cell, right? And you can come out and reassort into different
The reassortment is an advantage.
virulence.
20 to 50.
Yeah.
The pox viruses are 1 to 100.
Mm.
The worst one on
Yeah.
this table.
Sorry, Rich.
The worst are the papillomas, 10,000, 1 in 10,000.
That's incredible.
Alan, what were you going to say?
I was going to say that the way I got this table so quickly was I Googled particle to PFU ratio flu, and I found a blog post called Are All Virus Particles Infectious? By some guy named Vincent Racaniello.
Power of Google. And I was looking through my copy.
You were looking for your copy of a book you
I love this. I
chapter
love
you wrote.
this. I love this.
Beautiful. Isn't that great?
Google
It
beat you
is.
to
That's
it.
great.
How does the AIDS virus come out with that?
I just closed it. Alan still has it.
Too bad. That's right. He's committed it to memory. Alan, how does he? Just out of curiosity.
I don't think it's on the table.
Yeah, I don't think that's on the table.
Dickson, so, some viruses do the segmented thing, but there are advantages to that, as Rich said, reassortment, but there are disadvantages because you have to figure out a mechanism for packaging. So other viruses do different things, but they have other advantages and disadvantages.
Does packaging involve some host protein cooperation here?
I'm not aware of that. Okay? I'm not aware of any host proteins, but, if someone said you're wrong, I would believe it because it could be.
And
It makes sense.
Okay, fine. I won't.
It would make a lot of sense. What else did you want to ask?
No, I was just curious as to whether the fragments could recognize each other and assort according to families like 1, 2, 3, 4, 5, 6, 7, 8, rather than 2, 4, and 6 or something else. Maybe there's some association that these particles have for each other, which makes them packageable into genomes of 8.
That could work.
Yeah, so the idea is that there's some there's signals at the ends.
Yeah. That they can recognize.
Right, like 1 recognizes 2, 2 recognizes
Exactly.
3.
Exactly.
Yeah,
There is
like
actually
form a conga line and march into
Or
the passage.
elephants.
There is a phage that does that. There is a phage
Really?
with a genome of 3 RNA segments,
Yes.
and the first one goes in by some sequence, and then until that is in, the second one will not go in,
How about that?
and then when the second is in, the third will go
I'll
in.
be darned.
But as far as we know, that's not what happens with flu.
No.
But that virus, what do you think the particle to PFU ratio of that virus is? It's almost 1. Yeah, so there's no problem with packaging.
I'd say 1.
Anything else about this paper?
I'll be thinking about this all night.
I just wanted to mention, I think the experiment that Rich wanted with not cross-linking and looking at digestion is actually in figure 1.
I do have another question. The
And what's the result? What's the result?
The result is that there's too much degradation, and they can't really see anything.
Got it.
So this diagram here that I'm looking at from terminal NP loop,
All right.
it looks as though the proteins are in a specific order.
Which figure are you looking at?
In
the show notes?
Yeah, the show notes. Yeah. It looks as though each turn of the helix repeats in terms of the proteins that are stuck onto the RNA molecules. Is that true?
Well, the shape is helical, yes.
No, I don't mean that. I mean, is the order of proteins that stick to
They're all the same protein.
Oh, it's the exact same.
Yeah, NP is just repeated many times in the RNA.
Oh, I thought these were a series of proteins.
No.
It's NP complete.
So,
Sorry, that was an extremely nerdy joke, wasn't
that's sure. I was totally misled then. I'm sorry. It's just one single protein that coats the RNA.
Yeah, exactly.
But it looks so lumpy. It looks as though it has an order to the way it's distributed. That's all.
Well, of course, each protein has one end and the other end, and they interact with each other for sure.
But it looks as all right. Fine. I won't.
Should we do the paper over again with your new knowledge in hand?
I'll be totally quiet.
No, don't be totally quiet. It's fine. Many people probably had the same idea. You never know. And you represent
the uninformed public. This is exactly who I am.
I wasn't going to say that. Someone once said on TWiV, Dickson asks the questions that I'm always afraid to ask.
I'm not afraid to ask.
You shouldn't be afraid to ask any questions.
No, because I can think through a problem regardless if I know the details of why I'm doing this.
You can think through regardless of reading the paper. That's cool.
Well, I can't do that. But Rich admitted that he doesn't read them either. But he's a virologist. That's different.
You don't read the papers, Rich Condit?
He says he doesn't.
What? The papers we discussed?
Yeah, he said you don't read them.
Before the show.
I labor over these things. Yeah.
All right. I missed
it doesn't matter if you don't read them.
That's fine.
If you can participate in the discussion,
yeah, well, that's right.
Which you should be able to, because if you listen, you say, oh, what does that mean? You can just ask questions.
Absolutely.
It's fine. But I feel I should read the papers.
Right.
Because if no one says anything,
so
that would be awkward. Yeah.
If you change this protein a little bit by mutation, what happens then?
Well, you can change parts that contact the RNA so it no longer contacts the RNA. You can change parts that cause the interaction of NP with NP, and you can knock out.
Is that a strategy for controlling influenza then?
No, I don't think so.
What if you programmed the cell to make that protein that was slightly different, and it competed with the protein that the virus makes?
You can't give this to people. I think the best way is
to
make a vaccine against flu.
Yes.
And to make it so it hits all strains, and people are working on that.
Yes.
By the way, speaking of reading papers,
yes.
Yesterday we recorded a TWiV. Okay.
Oh, yes.
And earlier in the week, Alio had agreed to do the paper, the main paper.
Yes.
And when I dialed him up yesterday, he said, I forgot to put it on my schedule. I'm sorry, I have to go.
Oh, no.
So fortunately, I had read it, and I was able to do it.
Right.
That's why I read every paper, because you never know.
No, the question, though, relates back to a vaccine against influenza is antigen specific. And that varies with regards to the mutation rate for the RNA molecule.
That's correct. Yeah.
But this would not vary. The protein.
NP is quite conserved. That's correct.
So that would make a universal vaccine against this. But you could molecular.
It's conserved, but it's internal.
Yeah, it's inside the cell.
So this is inside a capsid, and then it's inside the cell, and then it's inside a capsid again. And so you're going to have something that's going to get you access to that for the antibodies to recognize it.
No, this is not an antibody thing. It's a competition thing. I was just thinking that you could create all defective virus if you could prevent these proteins from coding the RNA. So a defective.
A drug? You mean with a drug?
No, with a variant protein that you engineer the cell to produce.
You could do that in cells. Sure. I'm sure you can make a dominant negative NP.
Yeah.
But how could you give that to people? You can't give people dominant negative protein.
You'd have to infect their germline with it.
Not happening.
Yeah, I don't know if that's going to work.
I don't think that's a favorable cost benefit ratio.
But I mean,
you can't.
No, you're right.
You can't edit people, right, Dickson?
No. But scientists ask us questions anyway because.
But that works really well in cells for sure. You can make dominant negative NPs. Absolutely, most proteins that multimerize, you can do that.
Been done, right?
You would have to label those people as genetically modified. So
yes,
before they were eaten.
That's right. Before you sell them, that's right.
I'm not going to eat a genetically.
This person is genetically.
I can just see a joke about this in Cannibals, but we won't go there.
This episode is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. Blue Apron's mission is to make incredible home cooking accessible to everyone while supporting a more sustainable food system, setting the highest standards for ingredients, and building a community of home chefs for less than $10 per person per meal. They will deliver seasonal recipes along with pre-portioned ingredients so you can make delicious home-cooked meals in 40 minutes or less. And you can customize your recipes, get what you want on a time schedule according to your dietary preferences, of course. You get these wonderful recipe cards, which you can keep and use over. You get just enough of each ingredient so you don't waste any food. And you can choose every week what you want, or every other week, or once a month, whenever. Or you can let them choose and surprise you. Here's some upcoming recipes for September. Summer vegetable and egg panini with Calabrian chili mayonnaise and caprese salad.
Ooh.
Soy glazed pork and rice cakes with bok choy and marinated green beans. Skillet vegetable chili with cornmeal and cheddar drop biscuits. Pretty amazing, right? Now,
Yeah,
I bet...
that's good.
I mean, you're a good chef, Dickson, right?
I like to cook.
I couldn't make any of these, right?
Vincent,
And I
if you
wouldn't
can do
even-
lab work, you can do
and
this.
I wouldn't even want to go shopping. This is my part of cooking that I dislike
Right.
is shopping, because I don't like shopping and then I forget, and you have to get something. You buy a ton of it and then you have left over and it goes bad. But you get exactly what you need here.
That's
and the
true.
cool thing is, if you don't know how to cook, you can get one of these kits and learn, or you can teach your kids. I think this is a good practice.
That's very good.
Get your,
That's-
hey, let's make a Blue Apron
You know what?
tonight.
That's fantastic. I like
and
that.
get them involved in cooking.
I do like that idea.
And so it's pretty cool. It's got lots of upsides. There's no weekly commitments. You get deliveries when you want them.
Look at that.
And as I said, they reduce waste because they only ship as much as you want. They have a freshness guarantee. Every ingredient in your delivery is ready to cook, or they'll make it right. Now, I know that Brianne has done Blue Apron, right?
I have. In fact, after you read those recipes, I had to open up my Blue Apron app and see if I was getting them. Some of them sounded really good.
September recipes. Yeah, they're coming up.
I bet.
And Alan has tried it. I don't know, Rich, did you try it?
Yeah. I
tried it,
cooked
right?
several of these. Yep.
Yep. And Dickson
good.
was not.
No, I'm.
You like to do it on your own.
I like to forage in the grocery store.
You're a
Dixon's
forager.
mission is to serve man.
Ooh, I've read that book.
Check out this week's menu. Get 3 meals free with your first purchase with free shipping. Go to blueapron.com/TWiV. That's blueapron.com/TWiV. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron. So don't wait. Blue Apron, a better way to cook. Now we have a paper for you, which...
So is Blue Apron a good way to cook rabbit?
I've never seen rabbit.
Hassenpfeffer.
Hassenpfeffer. This paper is about rabbits.
It is.
Yes.
And it is a PNAS paper called Next Step in the Ongoing Arms Race Between Myxoma Virus and Wild Rabbits in Australia is a Novel Disease Phenotype. This comes from a number of institutions, including the University of Sydney, Pennsylvania State University. First author is Peter Kerr, and the senior author is Andrew Reed, and also on this paper is Edward Holmes. And we have previously done a paper of Andrew Reed's, which, and I forgot, I don't think I put it in the show notes. No, I did not. But that paper had to do with vaccination selecting for more virulent viruses, and we were very unhappy about that.
That was the Merrick's disease paper, right?
Might have been, yes.
I think so.
Yes, I've talked about that with students before, actually, and also feel very unhappy about it.
And this comes up again in this paper, as we'll see.
We have some issues here.
So, Rich, do you want to summarize the original rabbit experiment in Australia?
Thank you so much for asking, because I really do. I want to do actually 2 parallel stories, the discovery of the virus and the rabbit experiment. I spent an hour Skyping with McFadden yesterday to make sure that I was up to speed on this, and I had a bunch of questions about this. Okay, so let's talk about I'll try and be quick here. By the way, the reason we like this paper, the reason I like it, is it's all about host virus evolution. Okay.
Yes.
Right.
How all this stuff evolves. And it's probably the best sort of semi-natural experiment on this topic that's been going on for 60 years, almost 70 years now. Okay, so the virus. Go back to late 1900s. A scientist, an Italian scientist in Uruguay. You'll love this, Vincent. His name is Giuseppe Santarelli. Okay?
1800s, right?
1800s, late 1800s.
Right.
Okay? He's imported a bunch of European rabbits as laboratory animals, and he notices they start dying. Okay? And he figures out that it's a filterable agent that does it. It's not until later when a Brazilian scientist named Araújo, or I can't pronounce it correctly, figures out, characterizes the virus. It's a pox virus. The disease is called myxomatosis. He calls the virus myxoma, and figures out that this is a virus that's endemic in Brazil and infects a rabbit in Brazil that is called Tapeti. That's in the genus, the rabbit genus. Actually, they're Leporidae. It's in the Leporidae genus,
which I think has now
Silvolagus.
Yeah, it's Silvolagus now.
So yes, the genus is Silvolagus. The family is Leporidae.
But you're not telling us what the word myxoma derives from. I would love to know that.
Actually, I don't know.
Oh,
Why
did they choose that name if it affects rabbits?
It refers to it. I think its origins are. Alan can Google this, but the primary lesions, which I'll get to, look like tumors.
Oh,
okay,
right.
And I think that's one of the reasons that it was named this. At any rate, so the native species, Tapeti, a Silvolagus rabbit, is a different genus than the European rabbits that were dying in the lab like crazy, which is Oryctolagus. That's the genus for that. And so the story, by the way, it's a pox virus, so it's a large double-stranded DNA containing virus that replicates in the cytoplasm of infected cells. We've talked about pox viruses before. And they are masters of immunomodulation. They have a whole suite of genes that mess with the cellular and organismal immune response.
And myxoma, just to cut in here, is it's a general term for a tumor in connective tissue.
I see.
Okay.
I find that so surprising because myxo is mucus, I always teach.
Yeah, that's right.
Okay.
And this has nothing to do with tumors.
Nothing.
Right?
No, it causes, this virus causes growths that are, I guess, myxoma-like.
Yeah.
If you look at them right.
So, at any rate, the disease in the native Silvolagus species, Tapeti, in Brazil, is fairly mild. It's not nothing, but it's fairly mild. By the way, this is transmitted mechanically by mosquitoes. So the mosquitoes pick it up. It doesn't replicate in the mosquito. They just transmit it.
Call that a paratenic. That's
interesting.
Okay.
That's important.
That's the name of it.
Yeah, it's also important. May be important that yes, I think it's important.
That's because if you don't have enough viremia in the rabbit, it's never going to get transmitted because it's not amplified in the mosquito.
Right, and you got to have you have to have enough not viremia in the rabbit. Not only that, but in many circumstances, they're the highest concentration. Okay, so the disease starts out with these cutaneous lesions that are really, well, depending on the species you're infecting, can be pretty ugly. But they're these
pox-like.
Well, they look like they were mistaken for tumors to start with. Okay, so but they're these hyperproliferation and inflammation of tissue that gives you this lousy looking lesion. And they're not too severe in the native species. The infection in the native species can last for a couple of months, and then it resolves, and they're fine. Okay. In the European rabbit, which now we've already said is a different genus, you infect them, they get big, nasty lesions. It spreads. They get secondary lesions. They get a lot of bacterial infection. They get a tremendous amount of damage to internal organs. And it's one particle, one infectious particle in a rabbit is lethal. Okay. In a European rabbit. Okay. And by the way, for the transmission thing, the mosquitoes actually will bite the lesions where the highest concentration of virus is and transmit it to another animal.
Yeah, it's like, what is it, leishmaniasis? They bite the lesion, don't they?
Yes,
Sandflies,
right.
Exactly.
Yeah.
So you don't need a viremia to transmit.
No. Well,
when they
I guess
actually,
no.
Yeah, but when
You
they
could do
go
it
to the
just
next host and they take another blood meal, are they actually introducing the virus at the point where their proboscis goes into the skin of the rabbit?
I believe so. Yes.
Or do they bite them around the eye and then that's a mucus membrane?
No, you get them lots of different places.
Okay.
I think.
Okay.
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 All right. So, in the meantime, when the Europeans moved to Australia, they brought with them rabbits, a non-native species, for a couple of different reasons. Even in the first bunches of Europeans that landed in the late 1700s, they brought rabbits that they probably intended to use for food. But later on in the early to mid 18th century, there were people who reintroduced rabbits exactly for shooting, okay, for sport.
Yes.
All right. And, exactly.
To this day, Australia is a great place to hunt rabbits.
Certainly is.
So by the early 20th century, Australia was absolutely overrun with rabbits, non-native species, invasive species, and it was a serious problem. And in about 1950, an Australian pox virologist named Frank Fenner took advantage of what was known about myxoma and realized that this was highly lethal for the European rabbit. And he proved by experiment, including experiment on himself, that this virus wouldn't do anything to any other species, in particular humans. And so in about there were a couple of different early releases. The first one didn't take, but then 1951, they had a huge take. And I think there was another release within a year or so. And basically myxoma was introduced and absolutely trashed the rabbits. I think this was like over 90% of the rabbits.
So Rich, can I interrupt you just one moment here? This is a mosquito transmitted disease that doesn't require infection of the mosquito, but rather mechanical transmission. Mm-hmm. These rabbits were growing out in the middle of a desert. Where the hell the mosquitoes are going to come from?
I don't know enough.
I want an answer to that question.
I don't know.
Oh, the other question I have then is, can one rabbit just rubbing up against another rabbit spread it that way?
I don't know the answer to that either. All I know is that the apparently the predominant method of mode of transmission is mosquitoes.
There ain't a lot of mosquitoes in the outback of Australia. I'm going to tell you now there aren't.
Can I tell you?
There aren't.
The disease is spread by direct contact with an affected animal or by being bitten.
Thank you.
Okay.
Thank you.
There you go.
Thank you.
And by the way, the infection causes tumor-like growths on the skin. That's why they call it.
Right. Okay.
Yes.
But direct contact is another way of.
Yep.
Okay.
And that's important for this selection.
Absolutely.
So the first releases totally trashed the rabbit population because the mortality, case fatality rate is over 90% with this. It's basically 100%. Right. But then the rabbits started coming back. And this is because of resistance evolving in the rabbits. But that's not the end of the story. We're talking about 60s and 70s, and then the rabbits start dying back a little bit, and you wind up seeing viruses that are changed. That leaves us to this paper. So we're talking about coevolution of the rabbits and the virus.
Right.
Virus is really lethal. Rabbits get resistant.
Yeah.
Now what's the virus going to do?
Right.
And as you mentioned earlier, this is a good experiment because the idea is when a new virus is introduced into a new population for that virus, it can be extremely virulent. But over time, it may wane as the two populations, the virus and the host, learn to get along, right? But we really don't have any way to study that in humans because, all the viruses we have now, or most of them, were introduced so long ago that we can't tell what's going on. Even a recent one like HIV, say early 1900s, it's not even long enough to see anything happening yet. So we don't, in people, this is a hard experiment to do. But this one, we put it in. Yep. And we can say what's happening to it. And I would argue that it's still early days.
I would say so.
Oh, yeah.
And we're not at the end yet.
Well, and we put the virus into a species that is non-native to this place.
Correct.
And that's
so
another
it's really,
complication too, right?
But the whole thing is kind of a, as Rich said, kind of a laboratory.
Yeah.
Because the species is non-native and is multiplied out of control. So you have this system where the rabbits are not in the normal competitive environment that they would be in. And then you introduce the virus, which is done artificially. And so really humans produce this whole system. And now we know when it started.
Yes.
And so we can monitor exactly what's happening.
Exactly.
So this is all about the evolution of virulence.
Right.
Right. Now we get to the paper.
And I would argue that, so in the introduction, they talk about how, that initially, as Rich said, the virus was highly lethal for the rabbits, but then rabbits arose that were resistant. And as those arose,
or they existed in the population.
They existed. They were selected because rabbits multiply quickly.
Yes.
Lethality of the virus began to increase again. So it never went to zero. It did decrease. And the question here, as we'll see, is what's happened more recently. But I just want to frame this question. So the evolution of virulence, viral virulence, is a very interesting question. But I would say it's not framed correctly because in the evolution of viruses, the main trait, I would argue, is transmissibility. And that may affect virulence. But if you're just looking at virulence, you're going to miss cases where transmission increases without a virulence increase.
Right.
So,
right. And isn't that where the sort of direct versus indirect transmission issue comes in? Because you're going to affect virulence much more with direct transmission, whereas with indirect transmission, virulence and transmission have very little or much less of a link.
Right.
Right.
Now, what they say in this paper is that, well, if you get more virus, that's going to be accompanied by higher virulence. And I don't agree with that. Not necessarily true, because the native rabbits for this virus don't get sick from it. They probably have lots of virus in them, right?
There are no native rabbits in Australia. No,
not in Australia, in Brazil or wherever it was.
Oh, in Brazil, yes,
right.
Sure.
So that's my initial problem with this, the way they frame it.
Look.
They say, is there going to be an indefinite escalation of viral virulence? That's not what you should look at. You should look at transmissibility, viral titers that lead to that, and so forth. Right. So,
I think framing it as an escalation is also a mistake.
Agreed.
Because it's not that the virus is getting worse. I mean, the virus, when it was first introduced to the rabbits, destroyed the population. That's, you've got this incredibly high case fatality rate. And then as rabbits that could resist the virus grew out, then you had these susceptible hosts around, and now you've got another wave that we're going to talk about, where the virus has caused that population to fluctuate. But then what they're doing in this paper is actually taking the virus from various time points from the Australian rabbits and putting them into laboratory rabbits who are fully susceptible.
Got it.
Yeah, which may not be, but that's the way Fenner did it, though, right?
Apparently
well, I mean, in order to do the experiment and have a measure of virulence, it's perfectly reasonable to take some known uniform animal and test a bunch of different viruses in it. But that the virulence then is being, what you're calling virulence, is being described in that particular host, and it's going to be totally different in the rabbits out in the wild.
I totally agree with you. Yeah.
So we have a similar experiment that was done here, basically by introducing West Nile virus, right? And it was highly lethal for all kinds of corvides and robins and a couple of other genera of birds. And today, I think the crows have made a miraculous comeback. Basically, they're all over the place now, and so they've become resistant to this virus, basically. But I don't see the virus increasing virulence to kill off the crows again. So that might happen.
It doesn't have to, as long as it can transmit.
No, exactly
That's
right.
The point.
No, that's exactly the case, and I totally agree there.
So I'm always this study. All of these studies worry me because they're different from everything else that we see out there, right?
Yeah.
Where virulence is going up and down. I don't get it. I think they're looking at the wrong property and it may be in the wrong host, right?
Sure.
It's in a laboratory animal.
And the other thing they could say is that, hey, you know what? Our virus doesn't replicate in these mosquitoes, but yours does. So then maybe your virus is a virus of mosquitoes. And it's using birds as a vehicle to get from one mosquito to the next.
Right.
Whereas myxomatoma virus doesn't do that at all.
So what they did here was to get more contemporary isolates from Australia,
right,
and study them the same way that Fenner did.
Okay.
So he studied them in laboratory rabbits and New Zealand whites, right? And he put them into 5 virulence grades, and 1 is the most virulent and 5 the least virulent. So they did the same thing, and they got contemporary strains. They got 15, I think, different ones. And that's another thing. They picked 15 out of probably many, many. Who knows if they're the right quote unquote 15, right?
They also talk about, they say the viruses were chosen on the basis of mutations in known virulence genes.
Right.
That's weird.
Including reading frame disruptions resulting from insertion and deletion events and duplications of virulence genes. It says, and to be representative of the full myxoma phylogeny. Those strike me as being contradictory phrases, but nevertheless, there was some, according to that sentence, there was some selection.
There was a logic to the selection. They picked stuff that was mutated in presumed virulence genes. But it's definitely a specific selection of these viruses. They don't do one from every year or anything like that. And the
which would
the
be
selections,
better, right?
They kind of group bimodally. There's a 1950-53 time frame, and then there's a 1990s time frame. And I don't know why there's nothing more recent than 1999.
Rich, you mentioned before that the myxomatoma virus succeeds because it contains a cassette of genes that mess up the immune system. Are any of those genes amplified or changed to mess it up even more? So be
in
more
these viruses,
is that the basis for the virulence?
They don't know what the basis for virulence is. They have sequences on all these. We'll get to this. We're getting to it now. They have sequences on all these. There's one gene in common that's knocked out, but that can't explain things. They don't really know what's going on.
So I would say the selection of these viruses is suspect. They had some criteria, which I would argue is a bias. And like Alan said, maybe it would be better just to take one randomly from every month or every year or whatever. It's a lot more work, but why pick?
Yeah, and I understand you don't have unlimited funding and unlimited numbers of animals, but it would be nice to see a little more distribution in the selection and throw in a few examples that aren't quite along this same line, but they're clearly angling for a particular argument here.
But they have viruses isolated in nature, though, from the actual epidemic.
Yeah, these are all isolates.
Yes, these are all isolates
from rabbits.
Yeah,
and they have 2 from the 50s. They have a grade 1 and a grade 5. A grade 1 and 2 grade 5s from the 50s, and then all the rest are from the 90s, which the ones are more contemporary, and you want to know what's going on since the 50s. Right.
So the grade in the middle.
The grade 1 from 1950 is it's called SLS, and that's the starting virus.
Right.
Okay. And it has a high virulence. And then there's isolates from 52 and 53 that I presume that Fenner isolated for characterization, and they have a very low virulence. They're graded at 5. So that's the initial let me see this. Is that making sense?
Yeah, those are the ones that evolved to lower virulence. Not zero, but lower than.
Yeah, so
lower.
I suppose the logic is that it does, from the viruses, I'm being anthropomorphic here, but from the virus's perspective, it doesn't pay to be too virulent.
If you're running out of hosts, yeah.
Right.
So,
which they certainly were in 1952 to 53. And so the virus seems to have adapted, become less lethal, or you selected for a less lethal virus that could spread more easily between the more sparsely spaced rabbits. And then that stayed that way for some period of time. And then fast forward 40 years to the 1990s, and we start seeing these much more virulent strains popping up.
So there's one thing they mentioned a few times that I think speaks to your issue of transmission, Vincent, is that one of the important things about how virulent something is how quickly you kill the animal.
Yeah, right.
Mm-hmm.
Okay? Because the longer the animal lives with virus, the greater the probability that it can transmit virus.
Yeah, and if there are fewer rabbits around, you want them to live longer to transmit. So you could select for that. Yes, Dickson.
Oh, well, the curiosity is overwhelming here because, I see so many parallels between, let's say, cowpox and smallpox, and that the cowpox is not virulent, smallpox is highly virulent. And so why hadn't the cowpox become more virulent again, or why haven't the smallpox become less virulent again, or what's going on there with a different pox virus? But that's not my question. My question is, if I'm a low virulence virus, and I can infect all the rabbits without killing them, would the rabbits now become less susceptible to the virulent strain because the immunological machinery has been taught how to recognize a pox virus now? Do you fight off the higher virulent virus because you've already been exposed to the low virulent?
Do you become a naturally circulating vaccine?
Yeah, like for cowpox, like the milkmaids, and then smallpox. That sort of thing.
Maybe it depends on the population, right? If you have enough new animals born every year, that's not an issue. But like humans, good example, Zika has immunized enough of the population, so there's very little spread now. And so what we need is for new people to be born, but not that many are born every year.
And there are no carriers, I presume.
Of what?
Of myxomatomas.
Well, that's a good question. I don't know. Do you know, Rich, if they're carriers?
I do not know. I think the infection is resolved, but this is, nothing would surprise me. It turns out that there are poxvirus infections, in particular parapox virus infections, that look like they go latent or something and reemerge. So nothing would surprise me here. But I think the textbooks would say that it's an acute infection that resolves and you're done.
Is it mostly antibody-based immunity that you're looking at, or for pox viruses, just for pox viruses?
No, I think it's both.
Okay.
Well, I want to go back to just the original study because we can see what we're missing here. So the original virus killed most of the animals, close to 100%. Then it was replaced with viruses that had lower, sometimes 50% lethality. All right. And then years went by, the rabbits became resistant, and then viral lethality began to climb. And they said there are very few strains of virus that had case fatality rates of less than 50%. So it became more virulent. And we don't have any of those viruses in this study. We just have the original one, as Rich said, and then two that were less virulent.
Or the host became less immunologically competent.
That's possible also.
Right.
It might not be the virus at all. It might be the host.
Yeah, we're looking at partial information on two variables here. So the rabbits we're selecting.
They're not inbred. These rabbits are not inbred.
No. Well, you're absolutely right.
We're selecting resistance in the rabbits, but we're also selecting attenuation in the virus if it starts to wipe out all the rabbits.
Right.
And so we have a snapshot of two different time frames here.
Exactly. And you have a very large country.
And
this is an enormous country.
This is the best data we have on this problem, and it's still lacking, obviously.
The case fatality rates that you were talking about, Vincent, are actually measured. Those are measured in standardized laboratory rabbits.
Correct.
Okay.
Which are highly inbred.
That's all they have because you can't do it in the wild, right?
But the point is that you're using one model to measure the virulence.
You know what?
You could do this in the wild. Yes, you can do this in the wild.
You're right. You could get wild rabbits and do this, and you might find something totally different.
Well, it would be much more likely to reflect what's going on out there, don't you think?
If you caught 100 rabbits from one locale in Australia.
Right.
How similar are they going to be genetically?
I can tell you that. All I have to do is sequence their DNA. I can do that in a snap now. That's not a problem. You have a problem with that?
No, I'm just thinking, if you infected them, you might get some where you have severe disease, others that you don't. Maybe then you'd have to do genome sequencing and figure out,
it's easy.
Hey, we've been talking about that for a while on TWiV now.
You can take
humans.
those 100 rabbits and expose them to both strains of the virus and see which one's killing.
Right, let's see what they found, because we can talk about this for a long time.
We sure can.
I asked Rich, should we do this paper? Is there enough here? Well, there
this
you
is
go.
Enough for 2 shows, it sounds like.
All right, so most of the newer viruses, the 1990s, were grade 2 or 3 virulence.
Means high.
1 is the highest, 5 is the lowest. They had a few grade 1 and a few grade 5. Okay. And the strains from the 50s caused the typical cutaneous myxomatosis.
Right.
But all of their 1990s grade 1 to 3 isolates caused what they call an acute collapse syndrome. The rabbit.
I think this is the most interesting part of the paper, because
absolutely.
This is for sure.
So what happens is a few hours after infection, they get moribund and they die between days 10 and 15, but minor signs of the typical myxomatosis. You're not getting this cutaneous disease.
So its pathology is different too then.
Pathology is different.
My goodness.
Massive pulmonary edema with hemorrhaging, swelling of hind legs. No tumor-like stuff.
No time for that.
Bleeding.
No cutaneous lesions.
No cutaneous lesions, and they die relatively rapidly. And that is distinct from the original virus that was put in SLS, this is called, I believe, and some of the original viruses that spread from that. None of them do this, okay? They often found bacteria in the tissues, lung, liver, kidney, heart, spleen, lymph nodes, lots of bacteria, Staph aureus, for example.
Does this induce hemorrhaging?
It induces hemorrhaging.
Okay, so that's the mechanism of entry for the bacteria then, maybe?
Yep.
Well,
and low neutrophils.
Okay. Neutropenia.
Which are important for clearing bacterial infections, right?
Right. And wasn't it low lymphocytes as well?
Low lymphocytes, that's right.
Goodness.
Now, some of them didn't die quickly. Some of them took longer to die. And these animals had also secondary bacterial infections. And they suspect that these are very immunosuppressed animals.
All ages, both sexes?
No, all male.
Right. Mature adult rabbits, right?
Right.
New Zealand whites. Yeah.
So these new isolates are causing a very different disease associated with bacterial infection. The tropism is different from the original isolates, different kinds of tissues being infected. And bacterial sepsis seems to be an issue. They did some experiments where they treated rabbits with antimicrobials, antibacterial enterofloxacin.
Yeah.
These later strains from 93 that cause these acute collapse will survive longer if they're treated with an antimicrobial, suggesting that the death is in part due to bacterial infections, which I think is a really nice experiment. Right?
Yes.
Yeah.
They didn't have any survivors. Okay, the virus is still killing them, but it's not killing them as fast.
They have to do this in germ-free rabbits.
Right. They lasted longer, right? So the virus eventually kills them, but they last longer, presumably because the bacterial infections aren't bringing them down. And so the idea here is that these viruses are profoundly immunosuppressive. They're different tropism. They're infecting different tissues, but they're also immunosuppressive, which allows the virus to replicate and, of course, allows bacteria to get in. And that's basically the finding, right?
Right.
Yeah.
So what does this mean? Their proposal is that these viruses have been selected in the Australian rabbit population to be more virulent as a consequence of the increased host resistance.
It's weird.
I wouldn't put it that way.
I wouldn't put it that way. I would say that
these viruses have definitely been selected in the Australian rabbit population, but they've been selected in combination with them selecting the rabbits. And a result of that is that they have become more virulent in the laboratory rabbits.
Mm-hmm.
Right.
And it's definitely, as measured in the laboratory rabbit, a different a different phenotype. I mean, I don't think it's even necessary to talk about the degree of virulence. What really interests me here is that the pathogenesis is dramatically different.
Yes.
Yeah, that was what I thought too. Yeah, I would have loved to see, cytokine profiles.
Yes.
To look at what was happening, because perhaps there's actually a lot of interferon, and less of a cytokine that's involved in a
like
bacterial
immune
response.
damage. You're killing yourself with overreacting, you think?
I don't know. I wonder whether we're getting some immunosuppression based on cytokine responses.
I see.
Right.
So here's what they say. They say this myxovirus has evolved enhanced abilities to suppress the immune system of laboratory rats. Okay, so they do put that in there. We propose that viral genes encoding the acute collapse syndrome became favored by natural selection between the 70s and the 80s because they allow the virus to overcome the resistance of wild rabbits. And so they think this was selected for because of that. I think a good experiment would be to take some wild rabbits.
Yeah.
And put
absolutely
all of these viruses into them.
Or go back to Brazil and get some of those rabbits and see what happens.
Yeah, they haven't. These are the
that's effective. The Tapeti, yeah. That would be interesting as well.
They have an interest. They make a statement here saying we have no evidence that myxoma virus is evolving toward an innocuous fibroma type of disease as it induces in the Tapeti. But I would not even expect that. This is a different host. Why would you expect the same?
I got an idea. Let's write a grant.
Plus, I don't think you can, you can't really even talk about that without doing the experiment. Because I think that it's actually perfectly reasonable idea that if you were to take this virus and stick it in the Tapeti or stick it in a number of different wild Australians rabbits now, that you could get a phenotype, a disease phenotype that looked similar to the original myxoma in Tapeti. That may be what's happening.
Remember, Fenner's work was all done in laboratory rabbits also. So
sure,
you can say that in rabbits, this has a different phenotype.
Right.
But I agree.
Right. I think it's useful to do this kind of experiment and look especially with the historical comparison. You've got Fenner's data, and now you're going to do the same type of experiment with more recent strains. It's a great idea. I would like to know what some of the strains were that they excluded from the study, especially for those other time frames. Mm-hmm. But it's important to realize that what you're looking at is not the wild system. What you're looking at is an assay of something in the laboratory rabbit, and that shows you that these genes that have changed in the virus over the past 40 years do indeed have effects on virulence in the laboratory rabbit. Right. I buy that. What we don't know, and what we have no data on apparently, is what this is doing in the wild rabbit. And the wild rabbit, of course, has undergone this huge series of genetic bottlenecks as a result of this virus. So you've got that going on, and the wild Australian rabbit is probably quite different from the laboratory rabbit at this point.
Mm-hmm. Or from the introduced original rabbits.
Sure.
Right. And so now you've got a virus that is adapting to the situation in the wild, and it would be very interesting to see what kind of disease it's causing in the wild rabbits and how that has changed over time. But I gather that hasn't been tracked.
No. It could be that this immunosuppressive effect in a lab rabbit doesn't do anything in a wild rabbit.
Right.
And then all of this is not really meaningful in terms of virus host evolution.
Right. So
what is clear, let's return to the clear point, is that these more recent viruses have a markedly different pathogenesis disease phenotype in the laboratory rabbits. That's clear. That to me is the take-home message.
Totally.
Sure.
Okay, so there is evolution of the virus going on. How you want to label that, how you want to describe it, obviously is a subject of debate.
Right.
Well, you're only sort of measuring one side of this back and forth.
Yes.
And by the way,
two-sided battle.
By the way, Grant tells me that there are rabbit genome sequences being done.
Nice.
Of various rabbits.
So you
can see at a genetic level what some of the evolution entails.
So that last statement that you made, Rich, that this significance of this finding in the bigger picture of virus host evolution is unclear. They don't believe that because they conclude that our data show that a virus has evolved that is far more immunosuppressive, and this may have implications in agriculture and aquaculture, farm animals versus genetic engineering, immunization. They even say that it could lead to arms races in people as well. So I think they're taking it way beyond what the data really mean. And they say the last sentence, in the decades to come, it will be intriguing to learn which of the antagonists first fails to find a genetic solution to the selection imposed by the other. They are assuming that their results apply to the wild rabbits.
Right. And that there's an endpoint.
Right.
And that there is an endpoint. I think that's totally wrong.
Right. So I have a basic question about how far back in time were they able to go to get sequences on the early viruses? Did they get Fenner's original virus sequenced?
This SLS strain from 1950.
I think
yes.
Yeah.
So you should be able to show over time which part of the viral genome has been changing or been selected.
Oh yeah, they've got sequences for all of these,
and it's a region that affects.
It's not any one region.
No.
Well, it's
okay.
They're all different. They say in some of them there's not even any clear consensus as to what mutations might be responsible for this phenotype. So it's really a mix here.
It's a myxomatoma.
Yeah.
It's a mix.
Mixed up mytoma.
I can see how one possible explanation is that the rabbits get more resistant and the virus compensates for that by becoming more immunosuppressive, or at least that's the way it looks in the lab. Maybe even in the wild rabbit. I mean that makes a reasonable story, and that may be one. I think it's a kind of a simplistic way of looking at it, but it's a way of thinking about it.
Right.
It's a way of sort of packaging this whole thing, and that's fine. But I think you got to stop there. Okay. First of all, you have to say that that's one possible perspective, and you have to discuss the what's actually going on in the wild as we have it, and et cetera. But then these other statements about what it means for humans and vaccines and all that other kind of stuff really is over the top.
So I got a press release from Penn State on this.
Yeah, we have some bones to pick with it. Two different press releases here, right?
Yeah,
or is this all
two.
one?
I think it's all one from the News and Media Relations.
So this goes out to Ben Manning in the News and Media Relations Department at Penn State University, who's the contact on this press release. We've got a bone to pick with you, Ben.
So the first sentence of the press release: new research into a virus affecting rabbits presents serious implications about how viruses evolve, develop antibiotic resistance, and become more deadly. So.
Strike two.
Losing a lot of points on the exam there.
Steve, right two.
I don't understand what this antibiotic resistance. There's not even any antivirals in this paper.
That's right.
No.
Becoming more deadly. This is overstating. And I wonder if Reed even read this press release. However, he does have a quote here later on. He's quoted, "We need to be aware of this in areas like agriculture and even human vaccinations where we are artificially enhancing resistance," said Reed. "If our methods do not completely prevent transmission of the viruses, we could be accelerating the evolution of more deadly viruses." Now, as far as I know, vaccination of humans with a variety of vaccines has never led to this.
Never.
So
Right.
I find this very alarmist and inappropriate.
I would agree.
I would take it as an argument in favor of vaccines. We should make sure that we do completely prevent the transmission of virus by using our good vaccines.
The history of vaccination, yellow fever vaccine was fabulous, right? Still is. Same vaccine.
Polio vaccine.
Smallpox vaccine.
Look, the only one you might argue
vaccine.
is selecting for antigenic variation is flu vaccine, right? But then there's natural immunity that contributes to that as well. But
the rest of them work great.
They're not more virulent as a consequence.
No.
Right? So I mean, in a society where a lot of people are worried about vaccinations, I find that an irresponsible sentence.
And even in the case of flu, there's good evidence that the virus was shifting and drifting well before we had the vaccine.
Of course, absolutely.
But many vaxxers will grab onto this and run.
No, the vaccine didn't cause that. Yeah. So
crazy.
We had similar problems with Reed's last paper. If anyone remembers, this is really alarmist stuff. Overextending the conclusions.
This is part of a
paper.
pattern. Yes.
Well, and if you're going to talk about aquaculture and vaccines and all of those things, maybe you should look at more than one virus.
Yes, right.
Yes.
That's right. It's really dangerous to make broad conclusions based on one, and a study where we have issues with as well.
Exactly.
Right.
That's right. So over the years that we've been producing these vaccines, has anyone been monitoring the mutational changes that have occurred?
In what?
In which vaccines? Human vaccines?
Yeah, like say yellow fever vaccine, for instance.
Only
smallpox vaccine.
Well, smallpox, there's no more smallpox around.
Yeah, right? But you know damn well that viruses are changing all the time, randomly, and
we have
selected
we
for
have
randomly.
Right, let me tell you about polio. That's what I know. We've monitored polio viruses for years, and there's been no antigenic change. No change in virulence.
Got it.
It's always pretty much the same.
But the virus genome has had minor changes throughout it.
Oh, of course, always. Even without vaccination, it changes.
Right. But it hasn't affected
what that means. We don't know, but there's no antigenic change or virulence change as far as we know.
That's right. We've never had a failure of an active genomic vaccine for a given epidemic. Like, say, we've vaccinated everybody against yellow fever, but they caught it just now. What happened?
Right.
No, we've never had that happen.
No, the closest I think we have to anything like that happening, I guess, would be, pertussis. That's a totally different story. We switched vaccines, and we've got a less effective vaccine, basically.
And it's made differently too. So that's
and as a result, we're seeing a lower effective efficacy in the field, and that's causing the problem. It's not because the pathogen changed in response to the vaccine. I don't think we've ever seen an example of that happening.
That's not a molecular vaccine anyway. So that's right.
No.
Rich, anything else you want to say before?
No, that's it. I'm compelled by. I'm sitting here wondering about what this looks like in the wild, and I will bet you anything that what this is evolving towards is a situation like the original situation in Brazil.
Right.
We're 100, 200 years from now. I mean, it's already, the virus is endemic in the rabbits in Australia. 100, 200 years from now, we're going to have effectively an equilibrium.
So
as a matter of fact, it has not been effective enough, and there's still too many damn rabbits.
Yep.
I'm not going to blame.
They're looking at other. There was a Khaleesi virus that was
That's right.
being developed, and I think was either accidentally or accidentally on purpose. Released before approval at one point. And I don't really know what's happened to that, but it's still a problem. And who knows where the equilibrium is going to work out, except if you look at the individual host and virus, I'll bet you it's trending towards something like the original myxoma in Tapeti.
Well, what they should do is get something like Burmese pythons and release them into Australia. Right. And, throw a few other coyotes.
They've got some nice pythons over there. I don't have a bone to pick with Andrew Reed, to be honest with you, because I think all of us who write our papers would like it to be as significant as we can possibly make it sound. I have a big bone to pick with the reviewers for PNAS. They should have caught this stuff and said, you're hyperbolizing at this point. You're not allowed to do that. Stay within the data, stay within the research, and we'll publish your paper. Eliminate those sentences. That's their job, so they blew it. That's PNAS blowing it.
He put it there in the first place.
Doesn't matter. PNAS has the last say on this.
Okay.
They have the last say, and they should have told him, no, just like we did.
By the way, there was a more recent biological control effort in Australia. There were, I think, koi were overrunning their rivers because they got out of where they were supposed to be. There were floods and the koi got out. So they were going to put in herpes viruses to kill the koi. Now, I don't know what's going on with that.
It'll probably kill off the Murray cod before it kills off the koi.
So Australia has a history of trying to do this biological control, but I'm not sure it will ever work because of evolution, right?
Exactly.
Anyway.
So this issue of hyping your work, it's this is difficult because I understand the pressures. And unfortunately, hyping your work works. Sure.
Mm-hmm.
You know?
Yeah. And did Andrew Reid actually read what their publicity agent wrote about it?
That's a good question.
They rarely send it back to you for review.
It's a quote from him, but the antibiotic thing he should have picked up.
That's absurd.
That's crazy.
But just as a matter of principle, I would, I don't like it. Okay, I think we ought to stick to the science.
Let it stand for what it is.
Yeah.
Here's what we found.
Exactly right.
And if it really means something, then it'll go on its own, right?
Got it.
That's right.
Got it right.
All right. This episode is brought to you by the Defense Threat Reduction Agency. Imagine an everyday, inexpensive drone that you could buy online, modified by terrorists to spread chemical or biological weapons over a crowded football stadium or holiday parade. Plague, VX, sarin, weaponized influenza. Lots of questions about this. How would we prevent a scenario like this happening? How could we treat the victims? How do we counteract the effects? Well, there's a meeting that's happening this November which are going to address these and similar questions. Join us, Long Beach, California, November 28th for the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Collaborate with over 1,500 scientists, subject matter experts, military service members, industry partners, and academic leaders from across the globe who are committed to making the world safer by confronting chem-bio defense challenges. Part of the U.S. Department of Defense and charged with safeguarding our warfighters and our nation, the Defense Threat Reduction Agency's Chemical and Biological Technologies Department hosts this important conference and brings together the best and brightest from around the world. Please join us to share your important ideas. For more information on the meeting and to register, go to cbdstconference.com. You can also stay up to date with the meeting on Facebook. Search for DODDTRA, or on Twitter, CBDST Conference. The 2017 CBD S&T Conference, today's innovation, tomorrow's warfighter capabilities. All right, let's do a couple of emails. First one is from Ed Grow. Hi, Vincent and the TWiV Gang on TWiV 447. Pete asks several questions about ERVs, endogenous retroviruses, and I'll try to answer them focusing on human ERV biology, but drawing on examples from other organisms. If it were feasible to remove, say, 1 to 5% of the ERV base pairs from a sexually reproducing organism, would you expect that organism to be able to reproduce with an unmodified partner? That was one of the questions from TWiV 447. I think you might be alluding to Drosophila with respect to hybrid dysgenesis, which involves mismatching of autonomous p-element transposon-containing males with non-transposon-containing females. Resultant progeny from this mating can be non-viable or sterile. So far, I'm not aware of similar systems that give such a dramatic phenotype in humans as hybrid dysgenesis in Drosophila. However, you can easily imagine a system conceptually similar to the mutator system characterized in maize, in which a dominant allele called mu killer, essentially an inverted duplication of the mutator transposon, leads to organism-wide silencing of dispersed mutator transposons through the production of double-stranded RNA, siRNA's RNAi-mediated DNA methylation. Therefore, it is, of course, possible that some ERV sequences are maintained to serve as an inoculation or inhibitor of reinfection or recolonization of an organism by new ERVs. I'm not sure that removing interspersed ERV repeats would affect chromosome pairing and or recombination during meiosis, so in my opinion, it's unlikely this would lead to aneuploidy or lethality in fertility. But ERVs are thought to have mediated the expansions of clusters of duplicated and subsequently diverged crab-zinc finger genes on human chromosome 19 through illegitimate recombination, so it's plausible that they serve the useful role in expanding this class of genes, which in turn serve as an anti-transposon, retrotransposon ERV defense. This type of accident-turned-benefit seems to be common in host pathogen coevolution. Examples below. Although not leading to hybridgenesis per se, mice have a system of loci called FV1, FV4, friend virus susceptibility, which were initially defined as loci that conferred resistance to murine leukemia virus. When positionally cloned in sequence, these loci were found to encode portions of retroviruses which interfered with infection from exogenous viruses. For instance, FV1 encodes a GAG fragment that inhibits viral replication, and FV4, which encodes an envelope fragment thought to lead to resistance to receptor-mediated interference. So the evidence is clear in mice that ERV sequences can be beneficial, and this seminal work really inspired researchers to consider the possibility of beneficial ERVs in humans. Not to be too self-serving, but here are some recent examples in humans. Grow et al. Nature 2015. That's him. The premise of this paper was simple. Do ERV-K, which still retain the capacity to make retroviral proteins, and this is human ERV-K, become reactivated in development, or are they viral fossils that exist in our genomes but are largely inert? In this paper, we showed that HERV-K, which is re-expressed during pre-implantation development, can trigger an innate immune response in a cellular system, and that this can confer partial resistance to infection of exogenous viruses. Although most of these experiments involve boosting ERV expression level, as loss of function is tricky to do for dispersed polymorphic ERV loci, we think that ERV re-expression and turning on innate immunity in early embryogenesis might serve as a protective mechanism before adaptive immunity appears later in development. Maybe that's why ERV sequences are retained in our genomes and re-expressed during critical times in development, in essence using fire to fight fire. I think we did that or a similar paper on TWiV before. Then he refers to Chung et al. In Science, which we did in TWiV 382. Many ERV sequences are DNA binding sites for transcription factors that are involved in innate immune responses. In this paper, Ed showed that AIM2 is not as interferon responsive if you delete its ERV regulatory region. Apart from this defect, cells with the ERV deletion or the ancestral form with no interferon-inducible ERV would be predicted to be more susceptible to viral infections. Then we have a paper from Dorothy et al. Nature Genetics 2016. An older ERV called ERV-H primarily can't make proteins but encodes highly expressed long non-coding RNAs in human embryos and human embryonic stem cells, although there were some nice experiments testing its function in cell lines, which you covered in TWiV 279. It was unclear if these RNAs impacted human embryo development. This is one of the only examples in which true loss of function studies were performed for HERVs in vivo, which addresses Pete's original question. We started with a simple question: if HERV-H-derived long non-coding RNAs are essential for pluripotency in human embryonic stem cells or contribute to reprogramming of fibroblasts to pluripotency in induced pluripotent stem cells, do these HERV-H also affect pluripotency of cells in vivo in the embryo? After months of hemming and hawing, we convinced an amazing embryologist, Mark Wasidlo, to inject siRNAs targeting ERVs into human embryos. The cells with depleted ERV-link RNAs didn't contribute to the inner cell mass, the population of cells that will grow into the embryo, but instead contributed to the trophectoderm, which will form the placental tissues. These data indicate that ERVs can profoundly influence the developmental potential of early embryonic cells, and in some cases we have initial clues as to the mechanism to which they act. To recap, these three papers show that different ERV derivatives can provide beneficial functions for humans or human cells. So don't be so sure that ERV sequences are non-functional. But many ERVists, that's a new one for me, ERVists would reluctantly concede that the majority of ERVs are deleterious or neutral. Although evolutionary conservation analyses can help us triage which ERVs are maintained or subject to selection, the true test is loss of function studies, which can now, thanks to CRISPR-Cas9, be performed in cell lines, organoids, or even in vivo in some cases. Vincent and the gang, it appears you might need to do a Virology 101 for the ERVs. And since two of the best ERVists, Steve Goff and Paul B. Nash, are both in New York City, I bet you could make a damn good episode. But, of course, there was a recent paper where they took all the pervs out of pigs.
Right.
And the pigs are living. I mean, they haven't been brought to the opera yet, but
no,
we'll see what happens.
They haven't learned how to fly either.
Right.
Alan.
Okay, Justin writes, automatic jargon identifier for scientists engaging with the public and science communication educators. And he sends an article in PLOS One that, let's see.
It's a list of words that you
it's a list of jargon words that you can try to avoid.
Mm-hmm. Oh, these guys have actually developed a program that will look at your article and identify jargon words, and even I think rewrite some of it for you.
Yeah, it's a program.
Okay,
it's a program called Dejargonizer.
Nice.
That's very cool.
what's interesting is to look at the difference between what they call jargon and what they call, lay speak, I suppose.
Yeah.
Right.
Seismic event is an earthquake.
Yep.
Myocardial infarction is a heart attack.
Do they have upgoer?
Right.
Was that another term for a proctologist?
They didn't restrict themselves to the 1,000 most common words.
A rocket. Brianne's pick last week involved using common words. Was it 1,000 words that?
Yes. The most common 1,000 because 1,000 is not one of them. Words in the English language.
So you can't say rocket.
Yeah, they wrote about a rocket as an upgoer.
Yeah, that's.
And they have one of their
Randall
figures,
Monroe.
Right.
One of their figures here is just an example of how this works, going from a sentence that is uninterpretable for most people to something that, having been ground through this, is a lot more readable.
Right.
Dickson, can you take the next one?
You bet. Hello, Twix team. The AAAS gives an award for public engagement with science, and I think the team should nominate Vincent. So do I. Nominations are due by August 15th. The award is granted to scientists. Yeah, that's going to be a problem here. Maybe next year. The award is granted to scientists or engineers who get the general public interested in science, and to people who do that through basically any medium, including broadcasting. On the application page linked to above, the AAAS states that it's especially interested in people who reach out to more distant audiences and communicators who bring a relevant or complex message. I think the many TWiV episodes on emerging viruses or on outbreaks like the Ebola epidemic in West Africa and the recent spread of Zika virus count as both relevant and difficult. Those episodes addressed events that concerned a lot of people and went into way more detail than the news ever could. Hope you apply, and if you do, good luck. Rest, Bridget. Well, that was just $1 short and a day late. Sorry about that.
Well, it's okay, because I was nominated by Islam Hussein.
Excellent.
The guy who
terrific.
Viral vlog.
Nice.
So we'll see. However, I must say,
yes,
I have been nominated by others for the past 3 years
yes.
and have fallen short.
Well,
so it's fine. I understand.
Well, it's good to know that there's good competition out there.
Yeah, it's fine.
Here.
1 year, Bruce Albert's got it, so I can't argue with that. Right?
Yeah, that's fine.
Brianne, can you take the next one, please?
Sure. Ada writes, "Hello, Doctors TWiV. First, thank you for your informative and engaging podcast. I've been listening for several years now, and your podcasts never fail to liven up a run around campus or make a long road trip feel shorter. I'm writing to thank you for discussing the importance of organ donation and for encouraging organ donation on TWiV 455. I'm a student at UW-Madison, where I've been fortunate to get to see the hospital's transplant program up close. It's hard to describe the joy and gratefulness of a critically ill patient and their family who has just learned that a suitable donor has finally been found. So please continue encouraging your listeners to register as donors. One final plug in favor of donation. No matter how badly you may mess up during your life, I can state with anecdotal evidence that if you sign up as an organ donor, your mother will still cry for how wonderful you are when you die."
Boy, that's tough.
Yeah.
Little mixed message there.
Yeah. This brings up a related topic I hope to at least get the word out about. Some organ transplant programs are considering making use of imminent death donation, a process whereby a terminally ill patient can choose to donate before she becomes so ill that her organs are no longer useful for transplant. This topic is, of course, fraught with ethical issues, which would require much more time than I would reasonably want to take up in an email, so I would suggest directing interested listeners to this article, and she links an article in the Atlantic. Imminent death donation is of importance because it has potential to greatly reduce the number of people on the long waiting list for a transplant. This topic is also of personal importance for me, since my family has a strong history of ALS. So there is a high likelihood I or a family member will at some point face a diagnosis portending death in an average of 3 years, over which our bodies will become increasingly useless and debilitated. The idea of being able to make my own untimely death into someone else's miracle or second shot at life gives me at least a glimmer of hope. Anyway, thanks always for the fantastic work that you do, and keep fighting the good fight for science. Best, Ada. Not my real name, but I'd rather my name not be used since my family's ALS history is not commonly known among friends and colleagues.
Yeah, we got several emails about organ transplant, and this is
somebody tweeted to me as well, showing a page.
That was actually me.
That was you.
It was me.
Page from a textbook on the opt-in versus opt-out organ donation, and opt-out does definitely increase rates in countries that have it.
It's a pretty dramatic difference.
Yeah.
Wow.
All right, let's take one more. Rich.
Anthony writes, in Beyond Freedom and Dignity, Skinner notes that current students of philosophy are directed to read the works of the ancient Greeks. No teacher of physics today ever suggests that a beginner study Aristotle. Skinner's analysis is that the first questions asked about science were good ones, and so the field was able to progress. He goes on to say that the first questions asked about philosophy were the wrong questions, and so the field never gained traction. I remember asking my 8th grade science teacher if viruses were the original life form. His answer was maybe. I then asked what was simpler. He replied that was not the only thing to look at. Viruses might have originally been something complicated that went on to specialize as parasites. It's interesting that almost 50 years later, a time when the progress of biology could be compared to that of physics in the first half of the 20th century, ideas on the origin of viruses continue to travel in the same circle. Perhaps the questions being asked about the origins of viruses were the wrong questions. On a separate note, during the opening discussion of TWiV 456, Dr. Condit says that having left Florida, he thought that he was done with hurricanes. I might guess that you never thought that there would be a Sandy in New York and New Jersey. We live in interesting times.
We do.
Well, yeah, remember Sandy? That was pretty bad.
Who could forget? Well, there are some that did forget, like Ted Cruz, for instance.
Now, didn't Texas vote not to fund New Jersey for Sandy? That's what I heard.
That's exactly right.
Christie was ranting about that the other day.
Yeah, he was.
Why should there be a vote about it? Why not just help people?
They're idiots. Yeah. They're idiots. All of them are idiots.
All right, let's do some picks. Alan, what do you have for us?
I have an article that I guess this touches on an arc that we do occasionally here on TWiV about sailing.
No pun intended.
This is an article from Sail Magazine about a sailmaker. And now when I think of a sailmaker, I think of some people sitting around in a loft near the waterfront in Annapolis and sewing canvas into panels. It's not like that at all anymore. So this is a really cool technology article about how modern sails are made for racing boats. And as we've talked about on some other picks, this has become a very, very high-tech industry. What's neat here is that they essentially take individual fibers of either carbon fiber or Dyneema or aramid, these super plastics, and they put them together and they make tape. And then they have a robot that makes the sail out of the tape. It's just really a bizarre process, but this is the way that you get these super lightweight, strong sails that the ocean racers use. And it's got a whole series of photos of the process and how they shape the thing. Just really cool.
So there's no more canvas, right?
No canvas in these things. They're talking about doing some of them out of polyester for cruising sailboats.
Wow.
The cheap market, I guess. But the whole shaping process, they've got this massive table that changes shape in order to mold the thing into the shape that it's supposed to be. And by the way, this is all happening out in the desert in Nevada.
Right.
It's nowhere near the water.
No.
Because the waterfront property is all too expensive and they're sails, so they're going to ship them somewhere.
They'll test them on the salt flats.
So they're
boats
out
with wheels on them.
Yeah, they're out in the middle of the desert making these huge sails for ocean racing yachts out of tape, and it's really bizarre and interesting engineering.
One of these last pictures is of something that looks a lot more like a sail loft.
Yes.
It says, it's not all that mechanized. Human hands are needed for the final touches. The finishing floor was familiar territory at last. And I just noticed that down in the bottom of that picture, there's an actual sewing machine.
There's a sewing machine, yes.
Nice.
Yeah, cool.
Rich, what do you have for us?
What do I have? Oh, yeah, this is almost a Shaggy Dog story. So as part of my continuing effort to orient myself and educate myself about my new home, Austin, Texas, I took my wife and I took the grandchildren recently to the Bob Bullock Texas State History Museum, which is on the UT campus. Bob Bullock was a Texas politician who was in the last stages of his career was a very popular lieutenant governor, and he was also a history buff and seeded at least and did some of the fundraising for this museum. And it's actually quite a nice museum. And up in the display, up on the top floor of this museum, is what's called the David Vetter isolation suit. David Vetter was one of these bubble kids. He had severe combined immune deficiency. And there have been several highly publicized cases. He lived from 1971 to 1984. And because of his severe combined immune deficiency, lived in in an isolation chamber, basically a bubble for his whole short life, 12 years. To try and get him some more freedom, they made a spacesuit for him, basically. They got NASA to do this, which is why it's in the Texas State History Museum, because there's a lot of NASA function in Texas, and so the display is about this isolation suit. But that's not what got me. As you read the display, by the way, he wore it about a half a dozen times, and then he outgrew it, and they built him another one, but he didn't like it. He said, I think the whole thing was just a drag. Okay, so he stuck with his bubble. As a last-ditch effort, when he was about 12, they gave him a bone marrow transplant. They couldn't find a perfect match, so they used his sister. And he eventually died, and the display says that he died from a virus that came with his sister's bone marrow transplant that had been undetected. So for those of you on the panel who have not read this.
What virus do you think it is?
What virus was it?
Bone marrow. Is it from the bone marrow or not sure?
It was a marrow transplant.
HIV.
That was my first reaction, but no.
No.
Because that can be in bone marrow hematopoietic
excuse me?
Herpes?
What herpes?
You're talking to a non-virologist.
Cytomegalovirus? CMV?
Nope.
Wow. Well, it's not simplex.
HPV.
HHV.
HPV.
You're going to be kicking yourself when you...
Is it chicken pox?
EBV. EBV.
EBV.
Of course.
In the bone marrow, yeah.
He got mononucleosis, but died, get this, of a Burkitt's lymphoma.
Yeah.
Oh, my goodness.
It sucks. Yeah, so his sister had been infected, and it was just latent in her. Everyone's infected.
Right. Yeah.
So we wouldn't do this today because we knew he was immunosuppressed, and he could not receive a bone marrow.
Basically, after looking at this display, I ran home and looked all this up. I'm giving the wiki link on David Vetter as well.
That's nice.
So I thought
nice story.
It's a great story. Sorry for people who have to do this, but actually, gene therapy is helping these people nowadays.
Yeah.
That's right.
So, what gene would you give him for SCID?
I don't know exactly the genetic details of his.
Yeah.
Whatever he was missing.
Yeah. A lot of them either are missing one of the RAG genes, adenosine deaminase, or the common gamma chain.
Okay. So they're monogenetic, though? You can treat them?
Yes.
Very cool. Dickson, what do you have?
Well, I have yet another article from Cosmos Magazine, my go-to journal recently, and I uncovered this article that's absolutely fascinating to watch. I'm sure in the fall, and we're coming up on that time of the year, we can all go outside and see massive numbers of starlings and grackles and sparrows winding their way to a warmer climate, and they move as one. They're an enormous body of a single organism, basically, a mega-organism, and you wonder how they do that. How can they all know where to go? And when one turns, they all turn the same way, and the fish do the same thing. And so these people studying bird behavior have branched out into, they're at the Max Planck Institute for Ornithology, but they're also interested in just how do things know when to turn, when everything turns. And they decided to work on fish in this laboratory, and you can see them working on that. And then they will actually label each fish with a little chip. And then from above, they have various series of cameras. And
a lot of chips.
It's a lot of chips, a lot of fish.
And
they're
chips. Small, they're little
fish and chips.
Yes, the fish and chips, and it's little zebra fish. So they're these tiny little,
they're fantastic,
long fish.
But when you put them in a pure white tank with a pure white bottom, you can see everything. And so they've got these cameras, and they stitch it all together to make it into a single movie. And it's fascinating that not one fish determines the movement of the entire school. So if it moves in one direction, maybe one fish gives the signal, and if they move in another direction, another fish gives a signal. And they all follow because they don't know where to go, but maybe he does, so it will follow him or her, whatever. And it's just an amazing way of studying biology, I think. Like cohorts of populations. You're now watching the entire thing move. And they had some amazing pictures of, I'm sure you've seen this when the sardines move up along the coast of South Africa. Mm-hmm. And they show seals and sharks swimming right through the school. And there's a hole about the size and the shape of a shark that the fish make.
So cool.
It's amazing stuff.
This technology. He pours the fish into this tank, and they all swarm together to one quarter.
Right. Exactly right.
And then they can do a graphic. Oh, this is so cool.
So if birds of a feather flock together, so do fish.
I love some of the
I was amazed.
the nature parts of this video where they show herds moving together and stuff.
It's abstract art when the birds assume these weird shapes as they start moving back and forth.
I was sitting on the beach a few weeks ago, and I love just staring out at the ocean.
Sure.
And there are these little birds that fly about a foot over the water really fast in flocks of like 30.
Oh, yeah.
And I was like wondering, how do they know where to go? Because they come by every now and then, and they turn and they...
That's true.
It's the same idea, right?
It is exactly the same idea. That's right.
So cool.
Yeah. Glad you like it.
This is great. I like this very much. I want to work on fish tanks.
That's right.
You pour them in and watch them. That's very cool.
It's quite amazing.
Hey, Brianne, what do you have for us?
All right. I have something that I found this summer when I was looking for some models to help viruses. I came across a site that sells 3D printed viruses.
Oh, I've been looking for this for a long time. These are beautiful.
They are very beautiful. They are sort of in all sorts of colors, and they look very accurate. I'm always impressed with the things that I found, and I have a few of them in my office now.
Oh, you've just cost me a lot of money. I'm going to.
They're not cheap, eh?
Well, these are $30, $40 a piece.
They're based on the protein data bank structures.
They are. They're all based on the protein data bank structure, and they actually have not only viruses but protein structures and things like that. I actually had emailed them. Because they had a couple of small molecules, but not CGAMP, that molecule we talked about last week that we were working on in my lab. And within a couple of hours, they
could do anything.
Had it figured out to make it for me.
What is it printed? What is it printed with? What kind of material?
Plastic.
Plastic. Yeah.
It is plastic.
Now, why is it that when people write poliovirus, they always have to make it 2 words? It's 1 word.
Yes, it is. That's a good question.
Always, but these are gorgeous. Some of them look. The adenovirus looks a little weird to me. I'm not sure what that's about, but I'm getting a bunch of these. You could just cost me $1,000 because I want to cover my desk with it. And for teaching, these are great.
Them on your Christmas
they're perfect
trees.
for teaching.
So for every lecture, you could put in the front. These are the viruses we're going to talk about today.
Great idea.
Makes it visual.
Yeah, especially to talk about differences in structures.
Yeah,
right.
They're ideal.
So this is. I have a few printed I have a polio printed virus here, which someone gave me years ago. It's made out of clay. And I've always wondered, where can I get more of these things? And I've looked at 3D solutions. They're also so expensive, but this is great. Oh, and they have amino acids, nucleic acids.
These are cool.
Brianne, I mean, seriously, I'm going to spend a lot of money this weekend. And I'm going to get in trouble. Wow, that's awesome.
I apologize in advance.
Well, hopefully all our listeners will buy tons of these too, and this company will do really well. You know, you could advertise on TWiV, but you don't need to. You just got a free ad.
Exactly. You got a free ad, and somebody's going to spend a pile of money on you.
I'm going to spend a pile of money because I love viruses.
Yes,
absolutely. I want to marry viruses. I already did.
Good. Guess what? They already married you.
I already married viruses. All right. I have a. Did I cover everyone? Yes, I did.
You did.
So I have a pick, which is part of our arc on the sounds of silence. This is a parody. Confounds the science, and that's all I'm going to say. Just watch it. It is sung to the sounds of silence, and it is political, and that's all I will say, because sometimes people do not like when I get political. But Art Garfunkel played by Don Caron, and Paul Simon played by Linda Gower, from the Parody Project.
And they do a good job.
They do a good job. It's really good. And the lyrics are just...
And good
fabulous.
Costumes.
All right, TWiV 457. You can find it at Apple Podcasts, microbe.tv/TWiV. One of the best things you can do to help us is to subscribe. If you listen on your phone or tablet, you use an app to listen to the show, and just subscribe to it. There are even ways to subscribe on your desktop. We'd love it if, instead of just playing them when you want to hear them, just subscribe so they get downloaded. That really helps us a lot. It shows our numbers look good for advertising and so forth. And if you love what we do, consider contributing financially. We have several ways you can do that over at microbe.tv/contribute. And of course, questions and comments go to TWiV at microbe.tv. Brianne Barker is at Drew University. Thank you, Brianne.
Thank you. This was great.
And also at Twitter at BioProfBarker.
Yeah.
Now, I would guess that you could get lots of good material for your class by doing these, right?
I absolutely do. In fact, we talk about papers pretty often, and makes it a lot easier for me to think about papers to choose.
And I guess they could listen too and hear a paper dissected, right?
Yeah, they could, and don't always think about ways to criticize papers or think about new experiments or ways to extend the paper. So I like having them listen, and I like having them listen and realize that even people in the field don't know everything.
Well, today's paper in particular, often we don't get so critical or whatever. Today we did. And there's a paper published in PNAS, and it would be an eye-opener for them to say, oh, wow, there are questions about this, right?
Exactly.
It's pretty cool. Well, thanks for coming. Come back. We'll have you back whenever your schedule permits. You seem to enjoy the TWiV experience.
I do. It's a lot of fun. I'm happy to be here whenever y'all need me.
Great.
Rich Condit is an emeritus professor at the University of Florida in Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. It's great fun.
Dickson Despommier is at thelivingriver.org and parasiteswithoutborders.com. Thank you, Dickson.
You're welcome. This was a lot of fun.
And we're about to release in the next few months your course.
That's going to be even more fun. We're having more fun even doing that.
Yeah, we recorded Dickson lecturing parasitology, and Daniel Griffin is going to do it.
Right.
Going to be cool videos. Stay tuned for that. Alan Dove is at alandove.com. Also on Twitter is Alan Dove. Thanks, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the American Society for Microbiology for their support of TWiV. And I'd like to thank the sponsors of this episode, Blue Apron and the Defense Threat Reduction Agency. The introductory music on TWiV is performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.